All right, welcome. Today we are diving deep into a world you can't see, but a world that, make no mistake, runs our planet. We're not just talking about germs. We're opening up their secret playbook. We're going to explore the brilliant, ancient strategies that microbes use to survive, thrive, and, yeah, dominate. So let's get right into it. So, let's start with a mystery. This is the kind of question that keeps scientists up at night. A few years back, in Africa, a massive vaccination effort was on the verge of wiping out a really nasty form of meningitis. Total success story, right? But then, a new strain, meningitis C, just appeared out of nowhere. And here's the crazy part. When they looked at its genes, it was almost identical to a totally harmless version found living in a healthy person just a year earlier. So what happened? How does a harmless bug just flip a switch and become a killer? Well, the answer lies in what we're calling the Microbial Playbook. Just think of it like the ultimate guide to survival and conquest, a guide that's been written and perfected over billions of years. So today we're going to break down the 4 most important plays microbes use to run the world. And in the process, we're going to figure out exactly how that shocking transformation actually happened. Okay, play number 1. This is the most basic, most fundamental play in the entire book. It's all about brute force. See, microbes understand one thing better than anything else. There is serious strength in numbers. And they are absolute masters at generating those numbers at a terrifying speed. Their main strategy is this beautifully simple process called binary fission. I mean, this is efficiency at its peak. One single cell just gets a little bigger, it copies its DNA, and then it splits into 2 perfect clones. That's it. 1 becomes 2, 2 become 4, 4 become 8. You see where this is going. But here's the kicker, the part that really matters. It's the speed. For a common bacterium like E. coli, this whole cycle, what we call its generation time, can take just 20 minutes. 20 minutes. Now compare that to us. A human generation is what, 20 or 30 years? It's just no contest. And that speed leads to this. Yeah, you read that right. In just 36 hours, a single E. coli cell could theoretically bury the entire planet under a foot of its descendants. This exponential growth, this is their first line of attack. Before they do anything fancy, they just overwhelm you with sheer, unbelievable numbers. Of course, an explosion like that can't last forever. In any real-world environment, they follow this predictable curve. First, there's a lag phase where they're just getting used to their new home. Then comes the log phase. That's the crazy exponential growth we just talked about. After that, a stationary phase as they start running out of food, and finally, a decline phase. Understanding this cycle is so important because it tells us when they're most active and also when they're most vulnerable. Now, all of that frantic growth, well, it isn't free. It takes a colossal amount of energy. So how do they fuel this massive expansion? Well, they pull off a kind of energy heist on a molecular level. It all comes down to their metabolism, which really has 2 sides. On one side, you've got catabolism. That's all about breaking stuff down, like sugars, to release energy. On the other side, you have anabolism. That's using all that energy to build the new parts they need, like proteins and cell walls. It's this perfect continuous cycle of demolition and construction. And the molecule that makes all of this possible is this little guy, ATP, or adenosine triphosphate. Seriously, if a cell were a tiny city, ATP is the cash. It's the currency that gets spent to power absolutely everything, from building new parts to just swimming around. Catabolism makes the ATP, and anabolism spends it. Simple as that. Okay, so they have the numbers and they have the energy. But what directs the whole operation? I mean, how does a microbe even know what to build or when to divide? Well, that brings us to play number 3, the master plan, and it's all written in the language of DNA. Every single microbe has a genome. It is its complete instruction manual for life. For most bacteria, this is a single circular chromosome just packed with genes. And each one of those genes is a specific instruction for building something functional, usually a protein. This is the blueprint for everything that cell is and everything it can possibly do. But, how do you actually read the blueprint? It happens through a 2-step process called gene expression. First, in transcription, the cell makes a quick, portable copy of a gene. That's called messenger RNA. Think of it like making a photocopy of just 1 page from a giant instruction manual. Then, in translation, a little cellular machine called a ribosome reads that RNA copy and builds the exact protein it codes for. And that's it. That's how the DNA plan becomes a real functional tool. So far, the playbook seems pretty straightforward, right? Grow fast, make energy, follow the DNA plan. But bacteria have one more play, a secret weapon. This is the strategy that lets them adapt, lets them change their very nature almost instantly. And this is the key to solving our meningitis mystery.
It's called horizontal gene transfer, or HGT, and it is a total game changer. See, while we pass our genes vertically from parent to child, microbes can pass genes horizontally, directly from one cell to another. It's like they're trading cheat codes in a video game. And this is how a bacterium can gain a brand new ability it never had before, often in a single step.
So one way they do this is through something called transformation. When a bacterium dies, its DNA can break apart and spill out into the environment. Another nearby cell can then just slurp up a piece of this naked DNA and stitch it into its own genome. It's literally like finding a discarded blueprint page on the street and adding it to your own manual.
Now, a much more direct method is conjugation. In this case, two bacterial cells actually connect, physically, using this little bridge called a pilus. The donor cell then passes a copy of a small, circular piece of DNA, a plasmid, through the bridge to the other cell. This is basically the microbial equivalent of plugging in a USB drive and copying a file. It's an incredibly efficient way to share useful genes, like antibiotic resistance. And finally, there's transduction. This one involves a middleman, a virus. So sometimes when a virus is replicating inside a bacterium, it makes a mistake. It accidentally packages up a piece of the bacterium's DNA instead of its own viral DNA. Then when that virus goes to infect a new cell, it injects the DNA from the previous bacterium. It becomes an accidental genetic delivery service.
So here's the crucial point. Here's how it all connects back to our mystery. Through one of these methods, our harmless meningitis C strain picked up a whole set of genes from a more dangerous bug, genes that let it produce toxins and hide from our immune system. It didn't evolve slowly over generations, no. It acquired deadly new abilities in a single genetic transaction. It used the playbook to completely rewrite its own identity.
But the story doesn't end there. Because once we understood the microbial playbook, we realized something incredible. We could use their own plays, their own tools, for our benefit. We figured out how to hack the playbook. We totally co-opted their machinery. We used their restriction enzymes as molecular scissors to cut DNA exactly where we want. We use their plasmids as delivery trucks or vectors to carry new genes into cells. And we use other enzymes like DNA ligase as molecular glue to paste it all together. This right here is the basic toolkit of modern genetic engineering, and it's all borrowed from bacteria. And what can we do with this toolkit? Oh, just solve some of humanity's biggest problems. For diabetes, we take the human gene for insulin, pop it into bacteria, and turn them into tiny little insulin factories. For vaccines, we can get yeast to produce just a single harmless viral protein, creating super safe and effective vaccines. We've even engineered microbes that can literally eat oil spills and clean up toxic waste.
It's pretty amazing when you think about it. By deciphering their playbook, we've basically learned to speak their language. We've turned some of the world's smallest organisms into our allies, into powerful factories for medicine and environmental recovery.
And that really leaves us with one final thought. For billions of years, microbes have been the planet's master innovators. They've solved problems of energy, survival, and adaptation in ways we're just beginning to understand. We've only just started reading their playbook. So the real question is, what on earth is it going to teach us next?